the slide that we ended with at the end of class on Thursday. We're going to focus right now a problem. Well, let's see, what is the problem? Yeah, the problem focuses on NCX. This is the sodium-calcium exchanger. A secondary active transport uses the sodium gradient to pump calcium. So let's do this problem. Work with the person sitting next to you, or turn around and work with the people behind you. So the sodium-calcium exchanger, it catalyzes secondary active transport. It takes energy to pump calcium uphill, outward. You can make good arguments that the energy for calcium transport by NCX comes from each of the following. So the direct source of energy is the sodium gradient. But you can make arguments that the energy comes from ATP or mitochondria or glucose or the sun. You can make those arguments. Work with your seatmate or whatever, people near you, to come up with a one-sentence answer that argues for each of these. Let's take 5 minutes to do this. Yeah, exactly. The sodium potassium ATPase hydrolyzes ATP, phosphoryl hydride bond, being fancy about it, and makes the sodium gradient, which NCX uses to pump calcium. So Sophie's answer was as good as mine. Argument for mitochondria. Energy comes from mitochondria.
They synthesize the ATP that's needed for the sodium potassium.
Exactly. Mitochondria is the source of most of the ATP. Remember, oxidative phosphorylation provides most of the ATP. Glycolysis, a small fraction of the ATP that your cells need. So the mitochondria, oxidative phosphorylation makes the ATP that the sodium pump uses to pump sodium to make the sodium gradient that the NCX uses to pump calcium. Okay, good. Make an argument that this energy comes from glucose or other food molecules. The pattern should be clear. Anybody? Oh, not exactly. Right. So remember all that good stuff from self-phys. Glucose, glycolysis. I'm not going to ask you to remember for this course all the steps of glycolysis, but someday in comps you may need it again. Glycolysis, pyruvate oxidation, Krebs cycle, NADH, high energy electrons, all that comes from food. So there it is, just like Alexandra said, glycolysis, citric acid cycle, electron transport, oxidative phosphorylation, take the energy from glucose and turn it into ATP. Okay, somebody make an argument that this energy comes from the sun.
Photosynthesis, light and dark reactions, Calvin cycle, produces glucose as a plant, something eats the plant, and yeah.
Exactly. The sun takes nuclear energy and makes photons. Plants do all that stuff that Matt talked about, like dark reactions, so-called dark reactions, right? To make glucose or other food molecules that we animals convert into ATP. Okay, good. So that's energy flow. Okay, let's change things up here. Let's do some anatomy, some terminology of anatomy. Stand up, please. Let's do some anatomy. So I would like you to use your pointer, your right index finger, and point to the most superior part of your body. back. Rostral, or superior. Okay, so now point to the most inferior, or caudal part of your body. We don't have to bend over, just pretend you've got a ways to go. Okay? Even though caudal officially means tail, to the tail, this is not the most caudal part of your body. Your feet are the most caudal part of your body. Okay, take your pointer and point to your ventral surface. This is your ventral surface. Try to point to your posterior or dorsal surface. Dorsal, posterior. Okay, take your pointer and point to the midline, your most medial part of your body. Medial. Okay? Take your pointer and point to a lateral part of your thorax. Okay? Lateral. So medial, lateral. Take your right index finger and point to the proximal part of your right arm. Right index finger, right arm, proximal. Your index finger is the most distal part of your right arm. So you can't use your pointer to point to it itself. But if you, obviously, so this is proximal, this is the proximal part of the arm, and this is the distal. It makes sense, proximity and distal, distant. Words, those words in English come from these words in Latin. Okay, take your right index finger and point to your ipsilateral shoulder. Ipsilateral shoulder. Ipsilateral, same side. Contralateral, ipsilateral. You'll need to know these for neuroanatomy. Okay, so let's do some of the actions. So these are descriptions, right? These are adjectives. The nouns are going to be the names of muscles that we're not going to cover too much right now. Here are the verbs. So flex or extend are the verb versions of flexion and extension. Abduction, to abduct or adduct. And be careful here, there's only one letter difference, but they're real different movements. So here is the anatomically neutral position. So you might think that this, the most relaxed position is anatomically neutral, but it's not. Anatomically neutral is this, with your arms, thumbs rotated out. This is anatomically neutral. This is not, even though this is relaxed. Okay, go into the anatomically neutral position and flex your elbow. Flex your forearm. Okay, that's flexion. Now go ahead and extend it. Extension. Flexion, extension, or flex and extend. Flex, so that's easy. Flex your arm at the shoulder. This is flexion of the arm. This is not flexion of the arm. This is flexion. So if this is flexion of the arm, what is extension of the arm? Extension. So flexing the arm at the shoulder, extending the arm at the shoulder. Flexing the elbow, extending the elbow. Don't fall over, but flex your leg at the hip. Flexion of the leg at the hip. And extension. This is my extending the leg at the hip. Okay? So you'll have to get these verbs in your head because we will be using these words, and this is part of learning anatomy. Okay, now go into the anatomically neutral position and abduct your arm at your shoulder. That's abduction. And this is adduction. So with gravity, it doesn't feel like you're actually doing anything to adduct. Gravity is providing a pretty big assist. But you can imagine that there's muscles that need to pull this down. Imagine you're a gymnast and you're on the rings and you do this. That requires a lot of muscle. That's why gymnasts have a very strong upper body. Okay, we're not going to talk too much about rotation and pronation and supination, but they're going to be out there, so they're there. I'm putting them on the slide, but let's not do that now. Let's talk, so take your right hand, make a plane out of it, so rather than a finger, make a plane, and make a movement that suggests that you know which is the sagittal plane on your head. So, you know, this or this or this, the sagittal section of your head. That's sagittal. So sagittal, midline sagittal, that's a very special sagittal section. But this is a sagittal section. All of these are sagittal sections of the head, midline and then lateral. Medial sagittal section, lateral sagittal section. Sagittal. Coronal. This is the coronal section. So coronal, an anterior coronal section and a posterior coronal section. Coronal. Sagittal, coronal, and now obviously the last one, horizontal or transverse. Like this. So a superior coronal section transverse section of the head, and an inferior transverse section of the head. Okay? Sagittal, coronal, horizontal, or transverse. Language, memorization, anatomy. In lab, we will occasionally talk about muscle origins and muscle insertions. The origin of the muscle is the part of the muscle that is attached to the bone that doesn't move. And the insertion of the muscle is the insertion into the bone that does move. So the origin of the biceps is in the shoulder. And the insertion, here's the biceps, right? You know that. Origin of the biceps is in the shoulder. The insertion of the biceps is in the ulna. And so that was what allows the biceps to do flexion of the elbow. So in a hardcore anatomy class, you would be memorizing muscles and nerves that innervate them, and origins and insertions. Because knowing origins and insertions can help with knowing the actions. But I'm not going to get too busy with origins and insertions. But it is a thing. If you follow up with an anatomy class in PT school or med school or nursing school, it's highly likely that they will be asking you to memorize these things as well. Not just the name of the muscle, but what bones it attaches to. We're not going to do that too much. Okay, good. Some anatomy. Most of the anatomy in this class is going to be learned in the lab. Okay, so now we're going to turn our attention to some review of binding, receptors and binding. So here's an image from your text that indicates that we've got a cell that secretes a chemical messenger. This could be a neuron that's secreting a neurotransmitter, or an endocrine cell that's secreting a hormone. And then different cells have different receptors, some of which bind the ligand, and some of which don't. Right? So ligand is the general term for neurotransmitter or hormone. I'll use this language kind of interchangeably. If we're talking about the nervous system, we're usually talking about neurotransmitters, but neurotransmitters are a type of ligand. So we're going to do some binding, review from self-is. So here is the binding equilibrium. The ligand and the receptor form a ligand-receptor complex. And this is not an irreversible reaction, it's a reversible reaction. So if we talk about the association constant, when this reaction is at equilibrium, that is to say, when the forward reaction rate is equal to the reverse reaction rate, this is at equilibrium. And at equilibrium, the concentration of the complex divided by the product of the concentrations of the reactants is equal to the association constant for that reaction, forward reaction. We can look at this reaction in reverse. We can just say, okay, let's look at the dissociation or unbinding reaction. Here is the substrate or whatever. The reactant is the complex, and it is in equilibrium with the monomers, or the ligand and the receptor individual. So this is just the same reaction as this, only looking at it backwards. And now the Kd, the dissociation constant, is the product of the products divided by the concentration of the reactant. It's the same concept, only it's backwards when this reaction is at equilibrium. So those are, and I'll be honest, in this class we will mostly be talking about dissociation constants. I know for those chemists it doesn't make a lot of sense to talk about why would we focus on the reverse reaction? I'm not sure it's true for everybody, but for me, the reason I focus on the reverse reaction is that the units of the Kd is in molar, millimolar units, some sort of concentration unit, right? Molar times molar is molar squared, divided by molar is now units are in molar. Here, the units are molar to the -1. What on earth is an inverse molar? I don't know. That's why Kd, with units in the molar or millimolar or micromolar or picomolar, that just makes more intuitive sense. Plus, it's the Kd of the reaction that factors into the Michaelis Menten equation. So this is the Michaelis Menten equation, but not for an enzyme. This is a simplified Michaelis Menten equation just for a ligand receptor complex. So instead of Vmax, we have Bmax, the maximum amount bound. We don't have to worry about catalysis when we're talking about ligand receptors. So it's easier. So here's Bmax, here's the Kd, and here's the ligand concentration in the Michaelis Menten formulation, which is really just a derivation of taking these concepts and thinking about maximums and Kds. So this is an equation that I will ask you to use. I don't think I'll ask you to solve problems algebraically with it, but I might ask you to solve problems with it graphically. So here's the equation. If we were to graph this equation, well, let's just walk through it. If the ligand concentration, is 0, well then this term becomes undefined. But if there's no ligand bound, if there's no ligand, there can't be any bound. This term becomes infinite, so this term becomes 0. when the ligand concentration is very large, this term goes to 0, right? If the ligand is huge and it's in the denominator, then this quotient becomes very small. So it becomes vanishingly small compared to 1. And so the amount bound at a very high ligand concentration is high. Not only high, it is Bmax. It's approaching the maximum amount. And if you were to graph this, it would look, it's a rectangular hyperbola. Now let's take a very special case. When the ligand concentration is equal to the Kd of the reaction, then this becomes 1, this becomes 2, 1 plus 1 is 2, Bmax over 2. So if this is, here's Bmax. When we go to Bmax, 1/2 of Bmax, if the amount bound is 1/2 Bmax, we are at, the concentration of ligand is at the Kd. Okay, question. Which way do I draw this for a, so what is the units of Kd? It's concentration units. So it's either molar or micromolar. Well, let's just be consistent with this. Molar, millimolar, 10 to the minus third, micromolar, 10 to the minus sixth, nanomolar, 10 to the minus 9. When we get to hormones, we're going to talk about some that have an affinity of picomolar, 10 to the minus 12. So 10 to the minus third molar, 10 to the minus sixth molar, 10 to the minus 9 molar, 10 to the minus 12th molar. You're going to start to get good at just knowing these. You may already be good at it. So just to give you a sense, micromolar, so if the Kd is in the micromolar range, it's sort of midland affinity. So here I'm going back to the question I wanted to ask. Which way do I change this curve if I want to talk about a receptor that has a higher affinity for this ligand than this receptor? Blue. So I'm going to get a different color. And I want to talk about higher affinity. Joe?
It'll shift to the left.
What will shift to the left?
The KD.
The KD will be lower. The KD will be shifted to the left. A higher affinity ligand binding reaction. A receptor that has a higher affinity for this ligand would have a binding curve, and let's just say it has the same Bmax. It would have a binding curve that looks like this. Oh, that's bad. It's not very good either, but, and you can see, so if I'm going to the same Bmax, here is the 1/2 Bmax. There's the lower Kd. Higher affinity, lower Kd. So, if a receptor has an affinity for a ligand in the picomolar range, it's a very high affinity receptor. And obviously, so what would it look like for a receptor that had lower affinity? It's just the opposite. A receptor that, if we have a receptor with the same Bmax, but with a lower affinity, I don't want, this is harder to do actually. Well, no, that's bad. Don't do that. I'm not going to make it quite as low affinity as that. So you can imagine, to sort of imagine, that this is eventually going to get to the Vmax, but it's not on this scale. And so here is the 1/2 Vmax for the low affinity receptor. It has a higher KD. A receptor that has an affinity in the millimolar range is a low affinity receptor. Micromolar is sort of in between. So nanomolar and picomolar are considered high affinity receptors. Millimolar is low affinity receptor and micromolar is middling. Just so you know, so when I'm talking, just you'll come across these values. Those of you who have taken biochemistry or are taking biochemistry will learn about a transformation of the Michaelis Menten called the Scatcherd plot or the Lineweaver-Burke plot. Transformations of it's the same concept, but it's instead of plotting, ligand bound versus ligand concentration on a linear scale, it changes it, and things become different. And I'm not going to teach the Scatcherd transformation or the Lineweaver-Burke transformation, but those of you who take biochemistry will learn about it. Questions about KD, Vmax, high affinity, and low affinity? Other questions? Yes?
You said that micromolar is the middle point?
Yeah, I mean, not that it's the middle point, but just that a receptor that has a micromolar affinity is sort of neither high nor low affinity.
Right.
Okay, so let's go back to our blue receptor. Fix it. Okay, we're back to our blue receptor. Actually, we'll go to the next slide. Because now we're going to talk about inhibitors. Now, I didn't print this one out for you, but you'll have room on your handout to start a new plot the same as the one that you've just been working on. So everything is the same now. We've got a receptor, we've got a ligand, we've got a receptor. The receptor has a Vmax, and it's got a KD. But now we're throwing into the mix a competitive inhibitor. Somebody who remembers this from cell phys, describe the curve of this binding reaction, binding of the ligand, not of the inhibitor, binding of the ligand when there is an inhibitor present. What does it look like? A competitive inhibitor.
Vmax will be reached after a long time. So it's kind
Time,
of, so we need more ligand.
we will need more ligand to reach the same Vmax because they are competing, the ligand and the inhibitor are competing, that's the word competitive, for the same site. So eventually, we can get a ligand concentration that outcompetes the inhibitor and binds, and you get the same Bmax. So in the presence of a competitive inhibitor, I'm trying to draw this as a curve that's got one, I'm not very good at it. And this is not going, this is going to the same Bmax. If it's going off scale, but if we were to go to the higher concentration of ligand, we would get to the same Bmax. But now you can see that in the presence of the inhibitor, remember this is not a different receptor, this is the same receptor binding the same ligand, but in the presence of the competitive inhibitor, I drew that okay. The apparent KD, the KD that you measure, the apparent KD in the presence of the competitive inhibitor appears higher. The Bmax appears the same, it is the same, but the apparent KD in the presence of the inhibitor is higher. And that's how you know it's a competitive inhibitor, because it behaves like this. What does it look like if we have a non-competitive inhibitor, also known as an allosteric inhibitor? Inhibitor is not binding to the same site. It's binding to a different site on the receptor and making it allosteric, conformational change, making the ligand binding site different than it was before. Amelia?
So like the Bmax will be lower.
Yeah. In this case, at a high concentration of ligand, when the ligand is fully occupying its site and changing the receptor, and it's an inhibitor, let's just say we have a concentration of when the allosteric inhibitor is bound, at the concentration of allosteric inhibitor that we have, remember this is not the concentration of, the x-axis is still the concentration of the ligand, not the inhibitor. But let's just say we've chosen a fixed concentration of the inhibitor that knocks out 3/4 of the receptors. 3/4 of at this concentration of inhibitor, 3/4 of the receptors don't bind their ligand anymore. But the ones that do bind their ligand are the same as the ones that we had before. Right? They're not, those are the ones that haven't bound the allosteric inhibitor. So this is the apparent Bmax in the presence of a certain concentration of inhibitor. The curve looks something like that. Here is the apparent Bmax. Here's 1/2 the new apparent Bmax. Did this pretty good. The apparent KD in the presence of the inhibitor is the same as the original KD. So for a non-competitive inhibitor, or an allosteric inhibitor, the Bmax changes, but the apparent KD is not changed from the original condition, which is in the absence of the inhibitor. why do we have to do this again? We're going to be talking about inhibitors. Drugs, for example, that affect the serotonin reuptake transporter, SSRIs, are competitive inhibitors. Certain interactions of hormones and receptors are competitive or non-competitive reactions. So that's why we're spending this time reintroducing this core concept. Not reintroducing it, reminding people. Questions about this? So,
Can you repeat what you said about what is apparent and what it equals?
so in the non-competitive or in the competitive case?
In the non-competitive.
Okay, so in the presence of a certain fixed concentration of the non-competitive inhibitor, no matter how high a concentration of the ligand there is, some of those receptors have been killed, knocked out. They're not killed, but they've been knocked out by the binding of the allosteric inhibitor. So we have a lower Bmax, a lower apparent Bmax, when the non-competitive inhibitor is part of the mix. But it doesn't affect the apparent KD. And again, those of you in biochemistry will see how this behaves in the Lineweaver-Burke or the Scatcherd plot. All right, let's do a clicker and then we'll take a break. Do 2 clickers. Okay, if the cell expresses more of a certain receptor type, which binding parameter changes? One best answer. More of the receptor. The receptor is the receptor. More of it per cell, per unit, per minute. Okay, so I was trying to give you hints, but some of you changed from a correct answer to an incorrect answer, so that didn't help. More receptors means higher Bmax. If the receptor is the same, KD is the same. So A is the best answer. If you change the number of receptors, you change the maximum number that can be bound. But there were some questions about this because not all of you got it, and some of you got confused as I was speaking. What confused you, or what? Ask a question, Charlie.
So just to clarify, the Michaelis Menten plot itself is it's a per cell basis.
It's per cell, per test tube, per organ. It's something is defined in terms of the volume or the amount of the receptors there, right? So, oh, I see what you're saying. So in this case, I should have said per cell, right? Because some of you chose D because you thought, okay, other things could change and we're going to keep Bmax the same. No, but what I meant was on a per cell basis. And so that was the confusion, but that was my fault. So if you chose D because you were thinking on a per cell basis, there's the disconnect between my A and your D. Is that right? Is that good? Okay, so now, if cell A expresses a receptor type with a higher affinity than cell B, Which of the following is true? One best answer. Okay, good. I'm seeing a lot of correct answers. It's C. High affinity means low KD, and I haven't said anything about Bmax, so you can't say. Okay? So let's take a break, and we'll get to the two main types of receptors we're going to be talking about in this class: GPCRs and ion channel receptors. Let's take a break. Talking about ion channel receptors and G protein coupled receptors. I'm not saying that tyrosine kinases or whatever this one is, Jun kinase receptors are unimportant. I'm not saying that. I'm just saying that in this course, we're going to focus on these classes of receptors. So, briefly remember that an ion channel receptor or an ionotropic receptor, another name, so ion channel receptor, ligand-gated ion channels, ionotropic receptor, these are all synonyms for the same thing. Channels that are opened by a ligand. So here's a channel, right? Membrane protein. This is a blobogram of a channel showing that when a ligand, in this book it was called a signal molecule, when the ligand is bound, conformational change of the receptor, and then ions flow. We're going to talk a lot about these. And here's a bird's eye view of a nicotinic receptor looking down the channel. And here is a side view of a glutamate receptor, an ionotropic glutamate receptor. Glutamate is the ligand. The ions that flow are sodium and calcium. So in most cases of ionotropic receptors, the name doesn't tell you what flows, it tells you what opens it, the ligand that activates it. Acetylcholine, GABA, glutamate. So there's a glutamate receptor from the side. It's got an amino terminal domain, a ligand binding domain, so there's the gray spheres here are supposed to be glutamate or some other agonist. There's a new word. Ligands that activate receptors are called agonists. Ligands that inhibit receptors are called antagonists. Antagonists can be competitive or non-competitive. So here is, let's just say this is an agonist bound to the ligand binding domain, causes some conformational change that we can't really see, and some hole or pore is opened up in the transmembrane domain. We're going to know, we're going to learn more about glutamate receptors. They're the most important excitatory receptor in your nervous system. We're going to do a lot of that, but not today. Today, we're going to focus the rest of the class today on, I think the rest of the class, on G protein coupled receptors. So here is an image from your textbook of a G protein coupled receptor pathway that is coupled to adenylyl cyclase, protein kinase A, and phosphorylation. I dislike this image a lot because it is so bad. There's lots of things. We could spend a lot of time picking this apart. For one thing, well, so here, remember that this is a this is the blobogram. Here is the way a receptor actually works, looks. The membrane would be from about here to about here, so this is the membrane, this receptor is mostly in the membrane, mostly a transmembrane protein. The purple haze here is the ligand, bound to the ligand binding pocket. So this is the way this is a dopamine receptor. We're going to be talking quite a bit about dopamine receptors later on in the course. Here is a receptor I can't really see it here because of the lights, but let's look over here. You can see, so this is supposed to be, this is the membrane. This is the extracellular surface. Here's the intracellular surface of the membrane. Here is the transmembrane domain of the receptor. Here is the ligand sitting in the receptor in the pocket, the binding pocket. Here is in red is the alpha subunit of the G protein. This orange, it's hard to tell, this is the beta subunit, and this is the gamma subunit. The beta and the gamma stay together and stay as a beta-gamma complex. But here you can see the scale, the receptor, and the G protein, and it turns out adenylyl cyclase are all pretty much the same size. They don't, the receptor is not 10 times bigger than the G protein complex. So here is a slide from your self-phys book, which is better, a much better image of the receptors and the G proteins. And there's a blank here for you to work on with your classmates. Turn around, work with somebody, and tell the story of this receptor. So let's just start. I will get us started. Here is the receptor with no ligand bound. This little cup is supposed to represent the ligand binding pocket. It's not a very good representation of it, but it's okay. The ligand is not present. The receptor is empty. The G protein is it's tethered to the membrane through some lipid linkages, so it's a membrane protein, but it's not a transmembrane protein. The alpha subunit and the gamma, therefore the beta-gamma complex, are tethered to the membrane through lipids, covalently attached. The G protein is in the off state, which means it has GDP bound, and it could either be in interaction with the receptor or not. There's some equilibrium between the inactive G protein and the inactive receptor. Sometimes it's bound and sometimes it's not. Now, your job is to tell the story. What does this arrow represent? What happens? What do these red flashes represent? What's happening here? Tell the story that leads to this point of the G protein. Go ahead and work it out. Well, I'm getting tired of climbing up on the chair. Let's see if we can do this without me filling it in the box. So, what's the first step of the activation of this receptor? What did you write in the top of this empty box?
The ligand binds to the receptor.
Okay. There's another word that's missing from just binding. Diffusion. The ligand is wobbly, it's diffusing, it's not smart.
You change slides.
Question?
You change the slide.
Oh, did I gave the answer away? The ligand is just diffusing. It's bouncing around in the fluid, the extracellular fluid, and eventually, doesn't take very long, it finds a ligand binding pocket and it binds. The receptor is not a vacuum cleaner sucking the ligand towards it, it's just a receptor. And so there's diffusion and binding. Okay, then what happens? Joe?
Conformational change.
Conformational change of the receptor. That's what these exciting red lines are supposed to do. Conformational change of the receptor in response to ligand binding. Okay, what happens next? So,
The receptor is phosphorylated.
the receptor, no, we don't, so we're not gonna get to the point where receptors are phosphorylated. That's how we turn off receptors. That's not part of this story.
The G protein binds to
The G protein is also diffusing in the plane of the membrane. Let's just say it is bound to the active receptor, and it, kind of,
It undergoes conformational change.
it
to
undergoes a
dissociate-
conformational change.
Yeah, to dissociate GDP and dissociate GTP.
Notice the change in the GDP binding site, from binding GDP with high affinity to binding GDP with low affinity, due to a conformational change in the alpha subunit of the G protein. So conformational change of the receptor, interaction of the receptor and the G protein, conformational change of the alpha subunit of the G protein. Lowers the affinity for GDP, so what's going on here? Valerie?
Like exchange.
It's an exchange. GDP unbinds, and GTP, which is abundant in a happy living cell, binds to the nucleotide binding site. This is GTP binding. GTP binding to the alpha subunit is what turns it on. This is now an active alpha subunit. And it separates from the beta gamma complex. So it separates from the receptor, and it separates from the beta gamma complex. And the active alpha subunit, and sometimes the active beta gamma subunit, go on to do other things. So here's the story. I didn't include that, but ligand diffuses and binds. Conformational change of the receptor. Conformational change of the G protein. Dissociation of GDP and association of GTP. The GTP bound alpha is the on state, the active state, and it separates from the receptor and from the beta gamma. And both the active alpha and the liberated beta gamma can go on to do active things downstream. Molly?
What causes the activated alpha subunit to dissociate from the receptor and the beta gamma?
Conformational change associated with GTP binding.
So I know the activated alpha subunit is activated because it has GTP bound to it,
Right.
not GDP. Is there any conformational difference between activated beta gamma complex and an inactivated beta gamma complex?
The only difference between an activated beta gamma complex and a non-activated beta gamma complex is that this beta gamma complex is bound to a off alpha, And this beta gamma complex is free, is not bound to the alpha. Okay, so let's go on with the turning on pathway. Oh, so here's the pathway that your book describes. Receptor, G protein, GTP bound alpha is what activates the effector. The effector in this case, which is adenylyl cyclase. This is the GS story. It stimulates, the alpha S stimulates adenylyl cyclase. Adenylyl cyclase is an enzyme that converts ATP, which is also abundant in a living happy cell, into cyclic AMP. It's an enzyme. Cyclic AMP is a ligand. It's called a second messenger. So here's the first messenger, here's the second messenger, but it's a ligand that activates another enzyme, cyclic AMP dependent protein kinase, or pKa. And pKa, being a kinase, can phosphorylate things and change the cell. This is the GS story that I will ask you to know. If I asked you to know what self is, I'm going to ask you to know it in AMP. What's the GI story? What's the difference in the GI story? So there are receptors that are activated by ligands that are coupled to a G protein that is not GS. It is GI. What's the main difference between GI and GS? Matt?
GI, the inactivation of adenylyl cyclase.
GI inhibits adenylyl cyclase. So GI for inhibition. S for stimulation of adenylyl cyclase, I for inhibition of adenylyl cyclase. So if adenylyl cyclase is not turned on, what happens to cyclic AMP concentrations in this cell? Goes down. Why is that? It's because there is constitutive enzymes that break down cyclic AMP. So adenylyl cyclase makes cyclic AMP from ATP, and phosphodiesterase, which is always there in the background, is breaking it down. Cyclic AMP goes up when receptors are activated because this is now working faster than this. So let's do a clicker, and then we're not going to get to GQ, but we'll get to GQ on Thursday. So caffeine, some of you have dosed yourself with caffeine already this morning. Caffeine is it has two actions. It is an inhibitor of phosphodiesterase, and it is an antagonist of a receptor that is coupled to GI. What does caffeine do to your intracellular cyclic AMP levels? Those of you who chose A, tell me how you got that. Matt?
For the first part, if it inhibits PDE, and it's inhibiting the thing that breaks down CAMP, so you're going to have an increase in CAMP.
Right, so by inhibiting phosphodiesterase, it inhibits the breakdown of cyclic AMP, allowing cyclic AMP to build up. That's simple. Go ahead.
And then for the second part, if you're an antagonist of the GI-coupled receptors, that means you're inhibiting the GI-coupled receptors, which means you're inhibiting the G protein that inhibits adenylyl cyclase, which means you're inhibiting the inhibition of adenylyl cyclase, which means you're increasing the activity of
Allowing,
adenylyl cyclase.
exactly. So this is a concept that you're going to be seeing over and over again in this course. Inhibition of inhibition. Sometimes called disinhibition. When an inhibitory neurotransmitter inhibits an inhibitory cell, that can lead to excitation. In this case, it's molecules. Inhibition of the inhibitory G protein, well, of the inhibitory receptor. So, antagonizing an inhibitory receptor, inhibition of inhibition, can be stimulation. That's a common, we're going to see that again and again. Okay, so I'm going to ask you, it's weird. We're going to start on Thursday with the GQ story. So I'll ask you to review the GQ story before class so we can move quickly, more quickly through it.